Next speaker is affiliated distinguished Professor Emmanouil Dermitzakis. He is a professor of genetics, Department of Genetic Medicine and Development, University of Geneva Medical School, Switzerland. Emmanouil is interested in the genomics of gene expression and cellular phenotypes in human population and multiple tissues, and how this assists in the interpretation of regulatory networks and human disease variants. Today he's going to talk about population and personalized genomics to understand the human biology and diseases altogether.
Okay, well, thank you very much, and it's a great pleasure to be here again in this conference and in this university, and with colleagues that we interact frequently, either in person or from distance over Skype or on the telephone. What I'm going to try to talk to you about today is some advances that we've made through a particular project, but I'm going to sort of extend it a little bit on work that we've been doing in our lab over the many years, on how we can bring knowledge of genetics at the molecular level in order to be able to understand and interpret signals at the disease level. I think that's the... Yeah, okay. So, thinking about revolution in medicine, I think many of us are, some more than others, are thinking about how is the next revolution in medicine is going to go, where is it going to come from? And clearly, revolution in medicine is coming from advances in technology, all kinds of medical devices that allow for better interventions, better diagnosis, and so on. But one of the things that we have been missing over the many, many years is deep understanding of the human body, of how the human body works. And technology can go so far into being able to intervene if we don't really know how the human body works and how that intervention is going to have consequences on specific physiology and other aspects of the human biology. So what I'm arguing, and I think that's not a very difficult thing to convince, hopefully, is that the next revolution in medicine is going to come from deep understanding of human biology. There's many things that MDs do now, I'm not an MD, that are a little bit sort of blind. Many drugs, we don't even know specific mechanisms. They just happen to work and then we keep using them. And we only hope that there's no sort of long-term side effects. But better understanding of human biology is not only going to allow us to understand better what we've been doing so far, but also understand where we should be intervening with what kind of interventions, what kind of medications or even lifestyle change and so on. Now, one of the big problems that we have from the genetics perspective, and that's me as a geneticist, is that we generally look at this genome that we inherit at -9 months of our life, and what we're trying to do is we're trying to build relationships with organismal phenotypes, diseases that usually manifest themselves quite late in life, maybe when somebody's 40, 50, 60, even 70. And one of the ways that we've been thinking about it, and I think the most conventional way to think about it, is that there's DNA information that does something, and the reason that it's late onset is because it's the environment that is accumulating its effect over time that together with the genome gives rise to a particular phenotype. Now, I want to add another component of it that has been given less attention, which is the exposure to our genome. So we have been thinking of exposures to the environment as a long-term time-dependent effect. I think there's a good reason to be thinking of genetic variants as a long-term exposure that cumulatively leads to a particular phenotype. So, for example, if you think of it in sort of a naive way, if you have a hepatocyte that has, actually the genome has, but there's a mutation in the genome that affects specific functions of hepatocytes, you can imagine that it's a small effect if you look at it over an hour, but if that same thing happens, this suboptimal function happens over 30 years, that's going to have substantial consequences on certain cells. And it's even worse when you're thinking about cells that are not dividing, because there the cell doesn't have the ability to sort of reprogram itself or change whatever memory it has from this exposure of the suboptimal effects of functions that we're thinking about. So in a way, we have to be thinking the time-dependent effect, we have to be thinking more, not only in terms of the environment, but also in terms of the genome, and jointly, either by interacting or independently, how can they give rise to particular phenotypes. Now, the genetic information as well as the omics information that comes from, let's say, transcriptome, proteome, metabolome, and so on, has been given a lot of attention because people are interested in understanding that deep biology that I was talking about. Now, however, the genomic information, the genome itself, has not been able to provide the information to predict somebody's risk at birth. So it's very difficult to say, for example, whether somebody will develop diabetes on day one of their life. Now, we can somehow try to use this information in different levels without necessarily saying that the genome is not useful for prediction. And here's how to think about it. So clearly, understanding aspects of disease causality, including genetics but not exclusively, will give us clues about what kind of drugs we might be thinking of using, what kind of interventions we might be doing, as well as of environmental effects that might be relevant to, in terms of lifestyle changes that have an impact on the phenotype. That's something that more and more is sort of the intention and the objective for genome-wide association studies, when we're really not going to get the predictive value that might have been anticipated early on. But the second thing is that we can start really getting into individualized risk assessment, not from the point of view of birth risk assessment, but from the point of view of understanding somebody's risk much earlier than we can do it right now. So if instead of, for example, of assessing somebody's cardiovascular risk when we look at lipid profiles when somebody's 40 or 50, and when they get abnormal, then we can say that somebody's at risk, we can probably look at omics profiles like other types of metabolomics or transcriptome, combined, of course, with the genome, that gives us clues about this much earlier in life, which means that there's more opportunity for intervention. Sometimes lifestyle, sometimes pharmacological. Now, in order to put all these things together, we need to start, we can do it in two different ways. One way is to do this machine learning approach, where we just throw everything in some kind of computer, so this kind of black box way, and then learn something, which is going to be highly overtrained, highly specific to the data set that we have, and is not going to give us mechanisms and understanding of biology. The alternative way is to try to piece all these things together in a biological framework where we start to understand how these particular molecules interact, in which context, in which tissue, at which time point. The phenotype, I mean, the three alternatives would be that DNA is impacting the phenotype, which is what I'm arguing is the right direction. The other one would be that the phenotype is impacting the genotype. That doesn't happen. You cannot change your genotype because you get diabetes. Third one is that it's parallel. But it cannot be parallel because the genome is established much, much earlier than any phenotype of an organism, especially the multicellular complex organism of humans is established. Therefore, it's kind of like an axiom that we accept that genome to phenotype association is a causal relationship. Now, the specifics, we have to figure out which nucleotide and so on. So in order to start, we have developed strategies where we look for what we call expression qualitative trait loci, where we look at genes as phenotypes. So we measure gene expression from, in this case, RNA sequencing, and then we associate nearby SNPs, exploring the cis-regulatory landscape, or faraway SNPs looking at the trans-regulatory space. And the trans-regulatory space, you can imagine it as a SNP that impacts the transcription factor, and then that transcription factor, the levels being, let's say, modulated, impact the targets of that. So you can have an association like that I showed you before, where there's a correlation between the level of expression, in this case, and the genotype, and it's basically a regression, it's a correlation between the number of copies of C. But actually, the direction of that is kind of irrelevant. It's completely conventional. Now, why do we put so much effort and so much focus on looking at these expression QTLs? Well, we do that because about 90% of genome-wide association studies has been shown to reside in known coding sequences. And as Steve said before, 99% of our genome is known coding. So there's a lot of information there that is going to give us explanation for disease that we haven't been able to explore simply because we don't understand the known coding component of the genome. So here's an example. I have this region here. Well, I have a SNP that is associated with a disease, let's say diabetes. If you try to understand just very simple things, this is a SNP. What does it do? Just basic molecular information, what does this SNP do? The first thing to think about is that it does something to genes nearby. And the question is what and which gene? Okay? Because the SNP is here, there's no information as to what it does. And you cannot assume that it's doing something to the next gene 3', like for example this one here, because regulation doesn't work this way, it's quite complex. This SNP could be affecting actually a gene right down here, 2 or 3 genes away from the SNP position. So unless I know the mediator of that effect, the gene, I have no idea, I don't have a handle on the biology that is associated with this association that I found. So what we need to do is we need to understand what does a SNP do, and because it's most likely going to be regulatory, I want to explore how does it affect the expression of nearby genes. And there have been methodologies that try to integrate expression QTL studies with genome-wide association studies to ask the question, am I looking at the same causal variant or different causal variants so that I can use the eQTL as a hypothesis? But there's additional problems because genome-wide association studies are done on DNA and you just correlate an organismal phenotype. eQTLs are not done on DNA, they're done on RNA, and then the question is, which RNA from which tissue? There's more than 200 cell types in the human body. Of course, it gets more complicated, probably more than that. And so depending on which tissue you look at or which cell type you look at, there's going to be a different expression pattern. There's going to be a different effect of that variant on regulation. For some variants, it will affect one gene. For some variants, it will affect another or none at all. So the tissue that you're going to be looking at for RNA is extremely important. And also, what is important is from the perspective of the disease. If you're interested in diabetes, what tissue will you be looking at? Islets, pancreatic islets? Will you be looking at liver? Would you be looking at brain? What about other types of tissues? What about muscle or artery or heart muscle or whatever? Which one is relevant? And by relevant, I don't mean affected, I mean causal. So something that happens in that tissue predisposes somebody to have a cardiovascular event. This question is or a diabetic event, or getting diabetes. It's very, very difficult to assess what's the causal tissue because it's not something that is readily obtainable from the data. We cannot get human tissues just easily. I cannot just dissect every one of you and you remain living and get 30 or 40 tissues. And we need large sample sizes, which means that we need a systematic effort to be doing it. Just to give you one of these caveats, you can have a case where, again, bringing the same region that I showed you before, 2 tissues, pancreas and liver, let's say pancreatic islets in this case, they have the same eQTL, the same SNP, affecting the same gene in both tissues. But which tissue is mediating the effect of disease? Is it both? Is it one of them? It is important to know because if I want to understand network effects, I need to be looking downstream effects in the correct tissue, even if the gene is the same in both tissues. Or I can have this effect where the same SNP is impacting 2 different genes in 2 different tissues. Both appear relevant, liver and pancreas, but I'm not going to know which one unless I'm able to add additional information. So we need an unbiased way of looking at the human body rather than a biased way that looks at only what we call relevant tissues. And to do that, there's this project called GTEx, which was started a few years ago, which explicitly samples from post-mortem donors about 30 tissues, and the idea would be to get to about 900. We're midway now to 450. This will be done with RNA sequencing. There's whole exome sequencing, whole genome sequencing for the same individuals, and there's going to be additional assays, what we call enhancing GTEx that go into methylation, proteomics, DNA number sensitive sites, and all kinds of other things. So what we want to bring is all these tissues now in what I said, kind of a more unbiased setting. Again, it's not 200 cell types, but it's 30 or 40, actually close to 50 now, where I can discover eQTLs and I can start building these associations with genome-wide association studies and have a more unbiased interpretation scheme. This is the figure from the pilot paper, which was published last May, where you see the number of eQTLs by sample size for the 9 tissues for which we had a good sample size to discover EQTLs, which means above, I think this was 60 or 70. And as you can see, the numbers are, depending on the sample size, anywhere from 1,500 to 2,500 EQTLs per tissue. We started building some more kind of manually curated links between genome-wide association signals, like this one in blood pressure, with 2 genes which have not an appreciable level of expression in artery, in tibial artery as we get here, but actually a strong effect with respect to the EQTL relevance to interpret the GWAS study. But more specifically, we developed a few years ago a method where we statistically infer that correlation, and we can test that correlation between the GWAS and the EQTL to see if they overlap, if they're mediating the same effect. I'm not going to go into technical details, it's already published. And what we can do now is we can apply this in the 44 tissues of the GTEx project that we have available in this midway data, the 450 individuals. If you do that, you get a strong correlation of how many of the GWAS signals you can interpret, you can correlate with EQTLs, which is highly correlated with sample size. Even when you correct for the number of EQTLs that you have per tissue, because when you have more EQTLs, you're more likely to capture a signal. The reason that it correlates with sample size is because you have more rare recombinants, which disentangles the correlation of the genome. And when you do that, you can start getting this kind of patterns where you basically get a number of tissues here and a number of conditions here, and what you plot is the enrichment of the relevance of one tissue to one disease. So basically, where do I get an excess of interpretation, an excess of overlap of GWAS signals for, let's say, narcolepsy with some of these tissues? And you can start building hypotheses, not about individual signals now, but about tissue causality, the participation of individual tissues. I'm just going to skip this a little bit faster so you hear some of these profiles which are currently unnormalized for sort of the random expectation for 44 tissues for body mass index, type 2 diabetes, and schizophrenia. But just to give you an idea for what comes up, let's say in type 2 diabetes, you get adipose tissue to be quite enriched, tibial artery, and several brain tissues, which is not surprising, especially for almost all of them. Tibial artery is a little bit off, but we have to see what that means. And you can go one step further by saying that I can get the average contribution of tissues if I look at all variants discovered in the population, but not all of us carry the same risk variant for diabetes. So then I can ask the question, for the 30-40% of us that carry a risk, 30-40% of risk variants that each of us carry for a particular disease, where these are active. So you may find, for example, that for some individuals is this tissue, which is, I think in this case, is liver, or for other individuals it could be pancreas. Which means that although you can get a contribution in the population, you can also have personalized contribution of these genetic effects in tissues. Finally, you can use GTEx as a reference, where you have all these tissues, you have a few expression values for your cohort of interest with a lot of clinical information, and you can use imputation methods to infer expression patterns in tissues for individuals which you have for tissues you have never sampled. So you can get, for example, blood, skin, fat, some other easily accessible tissues like muscle, and get an expression profile which is not going to be perfectly accurate, but it's going to be close enough for brain, which you can never sample from a living individual. So to summarize, I think we have an unprecedented catalog of regulatory functional variants in the human genome. We get a first glimpse of these effects in diverse human tissues. We can turn this tissue activity into a tissue causality profile, and this tissue causality profile can implicate pathways processes, but also can become individualized at the level now of the tissue, not at the level of individual signals. So with that, I want to thank the GTEx Consortium and, of course, my laboratory and the funding agencies. Thank you.
Where are you?
Behind one of my students on top. Just a little bit. I am young. What do you mean?
All right. One question, please. One question.
There's one over there.
Yeah, I have actually just a few small questions.
Excuse me.
It's really, I think it's very complicated data. Do you have a sort of software or program designed just for as a bioinformatics for your project? This is one. Second, how far into the project, and how long do you think you need to come up with informative results that can be helpful?
I think the project is basically a long process. It's not going to end, in a year. We're going to get some clues in the next sort of few months. The GTEx project itself is going to finish at the end of 2016. We are going to launch. We're writing currently a white paper to launch an international GTEx project. It's one of the ways that we can. One of the values of the GTEx project is not just about the genetics of gene expression, but also the diversity of gene expression in multiple tissues given the diversity of environments. If you sample 900 individuals from the US, the diversity of environment is not going to be that much. But I'm pretty sure that if you sample individuals from the US, Saudi Arabia, China, Sub-Saharan Africa, Europe, you will get such a diversity of environmental effects combined with the diversity of the genome that you will get some pretty advanced picture of the human body biology and how it's affected by that diversity. So the idea would be to do this for 20,000 to 30,000 people worldwide. Now, for the bioinformatics tools, it depends on which level you want to automate that, or you want to do a research project, which means that you have to take pieces that you put together yourself. But there are some of these that we have produced packages for that we can share.
Right. As a courtesy for Steve, one more question for Steve.
So,
Yes.
just trying to understand. Go ahead.
Yes, please, Doctor.
Go ahead, yes.
Yeah, I'm a genome student. I'm still not graduated yet. And I have a question here.
Yes.
I was wondering about you said the human tissue types of QTLs, it's hard to obtain. I was wondering if you can use some tissue culture or maybe use some stem cells too, so we can obtain some of these tissues.
That's a very important question, very interesting question. Yes, you can, and no, you cannot. It depends on which way you want to see that. If you are thinking of sort of a purely genetic effect, and you had a perfect way to transform any sort of, let's say, stem cell or iPS into the appropriate cell type through differentiation process, you would get a better answer than you get here just for genetics. The disadvantage is that you get no environment because, by definition, the iPS should have removed all the environmental exposures of that individual, by definition. It won't, but then it would be random what has been left. So, it would be great to have a bank where we generate cell types from iPS, but that's not going to give us the full range of biological effects that we're looking for.
Okay, Steve?
So you're using GWAS data.
Mm-hmm.
Have you modeled how deep you can go on the type and number of variants that are influencing EQ2?
You mean number in terms of
Well, I'm assuming you're
frequency?
using GWAS SNP data.
Yeah.
I assume you're also doing CND, but could you go deep into multifactorial disease? Can you model 3 or 4 loci, for example, effective 3 or 4 loci?
Well, we can. That's the whole point of doing this at the tissue level rather than at the level of a single variant, because
Can you do it? You have an example?
we don't have an example because we haven't advanced that much, but that's the point. The point is to do this in a system-like fashion rather than in an individual variant, and I believe that the first level of that system will be the tissue. The second level will be the organ.
Right, okay, thank you very much for that. I think it was very interesting. Thank you.
Excuse me, one more question, please.
All right, okay.
What's the criteria used to determine the causal tissue? Like in diabetes, it's a syndrome. it's multi-organ disease. Okay, so
Yes.
how you decide that which is the causal tissue?
Well, there's not going to be one causal tissue. There's going to be multiple. The idea is that if I have, for example, let's say 100 variants for diabetes, which are known to be associated, I'm going to try to find in which tissue the activity is present that is also causal for the increased risk. And if I see, for example, a particular enrichment in one tissue, like you would expect for liver, for pancreatic islets, for brain, for example, then you would put a prior of that tissue to have a significant effect. And of course, that's going to be, in general, an average in the population that has a certain contribution, and it's going to be much more skewed at every individual because every individual has a different set of risk variants. I'm not sure if I covered you, but it's not an easy question to answer in the 10 seconds and in the practice.
Thank you very much. I think we're going to stop questions here. Thank you very much, Emmanouil. Now the floor is to the ladies section again, and this time with Professor Emmanouil, and the first time she visits and speaks in the IGMC. So welcome to her, and is from Shepherd Diagnostic Genetics here.